Okay, good morning, everyone. Thanks for joining Cardiac Sciences Grand Round Sessions. I'd like to thank Justin for picking up one of our sessions on really short notice. Justin is an assistant professor of the Department of Physiology and Pharmacology and Cardiac Sciences here at the Cumming School of Medicine. He completed his PhD in the Department of Physiology and Pathophysiology at the University of Manitoba under the guidance of Dr. Grant Pierce, where he studied the role of infection and stress responses in vascular remodeling. For his postdoc work, he joined the lab of Dr. Paul Cubes here at the University of Calgary, using imaging modalities to study the behavior and function of the innate immune system during infection and injury. He has contributed 27 peer-reviewed papers, including top-tier immunology-focused journals such as the Journal of Experimental Medicine and Immunity. And his future program in cardioimmunology focuses on understanding how the innate immune system contributes to normal heart function and how it modulates the healing response in the heart following infection and or injury. Pretty timely with all the stuff we've seen with COVID. He's going to give us a nice talk on the pericardial myocardial axis and post-MI cardiac remodeling. And thanks again, Justin, for picking up one of our sessions, and we're looking forward to your presentation.
Well, thank you, Carlos, and thank you for the opportunity to come chat with you guys today and share what we've been doing in the lab. I'll just share my screen. Everyone can see that, yes? Okay, so what I'm hoping to sort of go over today is, again, some of the work that we've been doing and that's ongoing within the lab, along with collaborators, with a focus on sort of understanding how the pericardium might play an important role in cardiac remodeling post-MI. And just a primer, I'll be giving this through sort of an immunological lens, being part of my research focus. So we all know that ischemic heart disease is a leading cause of mortality worldwide and is traditionally characterized by buildup of atherosclerotic plaque within the coronary arteries of our heart, which as it develops can impede blood flow, and in cases where the plaque becomes unstable can lead to blood clot formation and complete occlusion of these vessels. Now, the consequence of this is usually tissue death downstream of this blockage. And the heart, unlike many other organs, has limited regenerative capacity. So once you lose this muscle tissue, you are no longer getting it back, and your heart is then initiating sort of a constant remodeling process. And this process comes in different phases, but starts to happen very acutely after the actual injury itself or a coronary event. And so usually involves actual extension of the infarct shortly in the hours post-injury, which is due to cell death of the myocardium, as well as through necrosis and apoptosis, as well as spreading through protein breakdown. In addition, this continues over time where you get basically cleanup or tissue resorption, extracellular matrix degradation, as well as neoangiogenesis happening. And over time, this can contribute to sort of an adaptive remodeling phase where you do get some beneficial remodeling to at least keep the patient alive, leading to scar formation, but is also usually involves LV hypertrophy to meet the demands of the body. Over time, or in some cases, this adaptive sort of remodeling can become maladaptive, where the heart can no longer meet the demands of the body and you essentially enter into heart failure. So what we're trying to do in the lab is really understand sort of how do we get from these initial sort of remodeling points to developing either the adverse or maladaptive remodeling that leads to heart failure. And one area that's really garnered a lot of interest in the last probably 10 to 15 years is the role of the immune system in this process. And so we could see this very clearly experimentally So if we look at just the immune system in action, so this is a live imaging of the beating mouse heart. We could see that even under steady state conditions, you have immune cells of the innate immune system, so primarily neutrophils and monocytes that circulate through the actual blood vessels in the heart. And even if you induce just a very small injury, there's a rapid and robust response of these cells that come out of the circulation and enter into the injury site where they play key roles there. So what we're trying to do is understand sort of what they're doing there and what are the key players in this process. Now, when we want to apply this to sort of the myocardial infarction sort of system, experimentally, most people will use sort of a rodent model. So using a model called the coronary ligation, where essentially you tie a suture thread around the left anterior descending coronary artery, thus inducing a damage downstream of that. And so this can be done either in a permanent fashion where reperfusion is not done in a timely fashion, or in a reperfusion, ischemic reperfusion sort of setup. And what you end up getting is significant remodeling of particularly the LV, and in some cases, or in a lot of cases, mimics a lot what's seen sort of clinically. So this is used sort of as the workhorse, particularly in the experimental fields, before you move on to, say, pig studies before into patients. And so using these rodent models, the key player that's been sort of acknowledged as being a key modulator of that early remodeling process are cells of the innate immune system called macrophages. And so in the blood, these macrophages start off as circulating monocytes, and they circulate around your blood, as we saw in the video. And in response to injury, these monocytes, along with other immune cells such as neutrophils, basically get recruited into the damaged heart tissue and start to play roles in actually breaking down tissue by releasing proteases and things of that nature, but also taking up the tissue debris and also further promoting the inflammatory sort of cascade, so by releasing more inflammatory cytokines. Now, when monocytes enter into a tissue environment, they can differentiate into a macrophage. And these macrophages have the ability to polarize or change their phenotype depending on their environment. So as the heart starts to heal or turn past this inflammatory phase, these macrophages can then also become reparative in nature and are thought to contribute to angiogenesis, as well as extracellular matrix remodeling and dampening down the inflammatory milieu. So this has been sort of the story up until probably 5-10 years ago, and these were sort of the main driver in this response. But what we now know is that the story is further complicated by what we call tissue resident macrophage. So in each organ of your body is actually populated with macrophages that reside within that tissue under homeostatic conditions. So this, you can go through any organ and basically you'll find them there. And these were typically thought to be monocyte derived and they're constantly replenished. But what we've come to know is that many of these populations are actually seeded during development and are actually maintained locally. And they're thought to play important homeostatic roles. And the heart is no exception with cardiac macrophages being present there. And these macrophages are found actually in a high abundance within the cardiac tissue. So this is the mouse cardiac tissue, and we've labeled cardiac macrophages here in green. And what we can see is that they're actually present throughout the actual cardiac tissue. So this is just one slice of it. But basically they interact with the vasculature as well as are interdigitated with the cardiomyocytes. And some groundbreaking studies that have come out in the last few years have shown that these macrophages have important sort of homeostatic effects. So a few years ago, it was shown that these macrophages actually interact directly with cardiomyocytes and can actually help to facilitate or to fine tune electrical conduction in the heart, particularly at the AV node. And then more recently, just last year, it's also been shown that these macrophages are heavily important in actually recycling damaged and old mitochondria that are released from neighboring cardiomyocytes. And without these macrophages, maintaining your heart function is actually altered, as well as your electrical conductivity. So this has sort of changed our sort of our concept of where these macrophages potentially come from and how they may contribute to tissue injury. And so the revised view is that you have resident macrophages that are present under these conditions. You induce an MI, you get recruitment of new monocyte-derived macrophage. And at the end of this, during the chronic phase, you get sort of a mixture of both. And in very simplistic terms, these monocyte-derived macrophages that are recruited to the blood are thought to be more pro-fibrotic, so they contribute to more fibrosis, whereas the resident ones are thought to mitigate some of that fibrosis. So a balance between these two has been critical. And so this was the story up until about 2 years ago, before some of our work. And so when I was doing my postdoc, there's another colleague of mine, Jing Wang, who discovered, at least in the liver context, that in addition to monocyte-derived macrophage, is you also can get recruitment of other tissue-resident macrophages. And these are macrophages that reside in the peritoneal cavity, so our serosal cavities of the body, and they express this specific transcription factor, GATA6, that provides them their phenotype. And what you showed is that these macrophages actually can enter from the peritoneal cavity that surrounds the liver and can actually enter in the liver injury, and plays a key role there. So if you get rid of them, the liver has delayed healing, and so integral for this process. So I start to think that, well, the heart is also surrounded by a serosal cavity known as the pericardial cavity, which has long been sort of known or associated with sort of more mechanical support as well as lubricating functions around the heart. But if you look carefully in the literature, and it's relatively sparse, is you will find studies that have shown the presence of immune cells within this compartment, in addition to bioactive components, so cytokines as well as growth factors that are present under different conditions. The question is are these just a result of the cardiac environment basically filtering out into the pericardial space, or can any of these factors actually play a role and communicate with the heart in the reverse sense? And so I want to explore this more experimentally, and the coronary ligation model I had previously presented. What I didn't tell you is that in this conventional method is the parietal pericardium. So the outer layer of the pericardium is actually disrupted. So essentially anything that you would have in there has basically been lost and hasn't been accounted for in the studies to date. And so in collaboration with Daryl Belke of the Cardiovascular Physiology Core in the Libin, we've actually developed a new coronary ligation model where we actually keep the pericardium intact, and we send the suture wire across the outer pericardium around the LAD and then tie it off. So we can keep a patent pericardial space that we can actually collect and sample at different time points post injury to understand what's going on within that space. And so just comparing these two models together, we wanted to know whether it had any sort of impact on function. So it would lead us to believe that maybe this is something worth pursuing. And so what we showed is inducing the MI and looking 4 weeks out. So we compared disrupted pericardium in gray here and intact pericardium model in blue. What we can see through PV loop assessment on these mice is that we can see actually dramatic improvements in cardiac function, both in relaxation, so end diastolic pressure volume relationship, tau and dP over dt, as well as some, but not all, systolic function measurements, so preload recruitable stroke work, as well as dP over dt max. So indicating at least that the intact pericardium has an impact on maintenance of cardiac function post-MI. So this is quite a crude experiment, I'd have to admit, and then there's likely a lot of factors influencing this. So we wanted to dig in deep and to understand, okay, are there specific components within the pericardium or the pericardial fluid proper that are contributing to this? So trying to get around the whole potential mechanical or loading conditions that may have an effect. So essentially looking at these factors that are found within the pericardial space. And so the first thing we did was, obviously coming from an immune perspective, was to look at immune populations that are present within the cavity. And so we first performed pericardial lavage, which is not trivial in the mouse system. The space is quite small. But what we're able to find is actually a multitude of immune cells. And to our delight is that we found similar GATA6 macrophages that were previously described in the peritoneal cavity. And they make up the large majority of the cells in there. So they're the most abundant single cell type within the mouse pericardial cavity. And we took this one step further and actually compared these pericardial macrophage to other macrophages within the pleura as well as on the peritoneal, and subsequently also compared them to the tissue resident macrophages within the heart. And so this is an RNA-seq experiment where we've looked at full transcriptome analysis of these isolated cells. And what I hope you can appreciate is that the pattern that you see with the pericardial macrophage actually mimics what we see with the pleural and the peritoneal macrophages, and is actually dramatically different with the expression pattern we see with the cardiac macrophage. So this would tell you that their phenotype is dramatically different. So even those cells that are basically sitting right beside each other in different compartments have a completely different expression profile, which would indicate they probably have different roles. So this is all fine that we show this in the mouse, but does it have any relevance to the clinical context? And so at this time, I sort of reached out to Paul Fedak, who had been doing some more sort of translational work, and I asked him whether he'd be interested in looking at this in some of his patients. And so he sort of linked me up with his PhD student and cardiac surgery resident, Dr. Ali Fateh Hassanabad. And we sort of started doing the work of actually trying to characterize this in the human context. And actually, to somewhat our surprise, is that we actually found very similar immune profiles to what we saw in the mouse. And we also found GATA6 macrophages that, again, represented about a third of all immune populations there. So we thought maybe we were on the right track. And this is something that might have important clinical relevance. So we decided to dive in a little bit deeper. So then we asked, okay, so what happens to these GATA6 macrophage following injury? And what we found is when we induce the MI, again with the intact pericardium model in the mouse system, is we see very early a disappearance of these cells. So they're no longer found within the pericardial space, and they seem to rebound. The population seems to start to rebound at a later time point. So does this also happen within the patient context? So again, collaborating with Lee, we decided to sort of look at patients in sort of two different groups. So these are patients undergoing cardiac surgery where we're collecting the pericardial fluid, and we've basically split them up into two main groups. So a non-ACS group, so these would be isolated valve cases, so either repair or replacement. And we're comparing these to an ACS group. So these are all CABG patients and limited to unstable angina, NSTEMI, and STEMI diagnosis. And these are isolated cases, so they're not a combined valve sort of combo. And what we did is, obviously we can't dictate when we actually collect fluid after the patient has suffered their event. And so what we did is we stratified the patients we did collect based on days the fluid was collected, or days after the event when the surgery actually occurs. And so we're able to look very early in the early inflammatory phase, so within the first 3 days, the proliferative phase, which is days 3 to 14, and then beyond that is the maturation phase, which is more reparative or long-term chronic condition. And what we found is actually very similar patterns to what we saw in the mouse. So in GATA6 macrophage, very early in the first few days, you actually see a decrease in these cells or disappearance of these cells, with a rebound as you move further out after the acute event. And just to compare it to two other populations that are heavily populated within this environment, that didn't show any differences between these groups. So there's something that's happening with these GATA6 macrophages that is likely important. So pursuing this further, we had the hypothesis that these cells can actually move into the heart and may be able to be recruited in there, similar to what we had previously seen in the liver context. And so for this, we went to some imaging where we used reporter systems where we can tag these macrophages with fluorescent proteins. And so you can see under baseline condition, if we take a snapshot of the epicardial surface of the heart, so the large majority of it, and then a zoomed in image, you can see the odd macrophage that interacts actively with the pericardium. If you compare this to a heart that's been has received an MI, what you can see is a massive sort of recruitment and coating of the epicardial surface with these macrophages. And if you take a deeper dive into this, so using some more imaging techniques, what we can see is that these cells that seem to line up with the mesothelium actually start to move in beyond this sort of mesothelial layer and epicardium, and looks like they're starting to move into the myocardium sort of proper, in specifically the infarcted region of the heart. And so right now we're designing ways to track these even further to see if they further integrate into the tissue. And this is sort of ongoing work. So we know they're present within the cavity. We know they're disappearing and sort of relocating to some extent within the heart. The ultimate question is, do they have any sort of impact on cardiac function? And so what we did for this is we used a sort of a genetic approach where we're able to deplete these GATA6 macrophages specifically. So we get rid of about 75% of them within the cavity. And we compare this to a littermate control. And so when we do this, we already see trends with more severe drops in ejection fraction when you're comparing the knockout to the wild type condition. And if you look at PV loop assessment to compare more closely with the disrupted pericardium and intact pericardium comparison I previously had, what we see is one of the major hits that we saw was that in the absence of these pericardial macrophage, you actually see development of a stiffer heart. And we're also seeing some trends in some of the systolic function parameters. So we're not fully recreating what we're seeing between the disrupted and intact pericardium models, but this is getting rid of one single cell while keeping the environment the same. And we're still able to initiate some of these events. So that tells you that these cells do have an important role in the actual myocardium proper. So because cardiac stiffness was a key readout that we had seen, we then pursued whether this was due to changes in actually fibrotic development within these hearts. And so we first looked more overtly or globally at fibrosis within these hearts. And what we can see is that the LV wall is heavily fibrosed in this model, but not major differences on the level of fibrosis on the outer wall. But if you then focus in on sort of the remote area, so the remaining viable myocardium, what we do see is increased interstitial fibrosis in the context of absent pericardial macrophage. And so we've looked at this both with serous red as well as collagen stain. So we've started looking at this in greater detail and trying to understand sort of what drives these macrophages to control this process. So we know that fibrosis is typically thought to be driven by activity of myofibroblasts that have a large capacity to produce extracellular matrix. And although they can derive from many different sources, their main source is typically cardiac fibroblasts that are found within the actual heart. So the question we had is, can these GATA6 pericardial macrophage directly affect the ability to generate myofibroblast from a fibroblast source and ultimately changing this fibrotic activity? So again, collaborating with the PhD lab, with Ali, what we've done is we've taken human cardiac fibroblasts and seeded them on collagen matrix. And when you stimulated them with TGF beta, they sort of take on this like spindle shaped, not spindle shaped, but sort of dendrite heavy cell type that is indicative of a activated fibroblast into a myofibroblast sort of phenotype. What we saw in these initial experiments is if you incubate these with pericardial fluid immune cells in these same conditions, what you see is that you get much less activation. So indicating that it's likely affecting the phenotype of these fibroblasts or their ability to actually go into this sort of activated state. We took this sort of a step further using another assay looking at fibrotic activity. So this is called the gel contraction assay, where you basically load, again, a 3D collagen gel with fibroblasts. In this case, these are human cardiac fibroblasts. We load them up. If you stimulate them with TGF beta, you activate these cells and they start to actually pull on the collagen matrix in there and they cause the gel to contract. So what we found is that compared to just the fibroblasts alone with the TGF beta is if you then add the pericardial fluid immune cells, you actually get a reduced amount of contraction, indicating they're less activated. And if you specifically enrich for GATA6 pericardial macrophage, you get that same effect, suggesting that these are the main cells that are providing this beneficial effect. So this is sort of ongoing work, but what we've shown so far is that we've been able to sort of establish or define a new cell type that's found within the pericardial cavity, and then in response to ischemic injury can actually migrate into the heart and have a role in sort of mitigating interstitial fibrosis. And so we think this is through potential modulation of fibroblasts. But it's still really unknown how it's actually doing this and the molecules it's actually making. So we're making some headway on this, but this is sort of an ongoing process. But what this initial project has really shown is the impact of how the pericardium can actually affect what's happening within the heart itself, and sort of motivated me to see, okay, well, can we do things or can we target the pericardium to actually further influence what's happening in the heart in terms of cardiac inflammation and remodeling? So can we look at altering what's happening in this space to actually affect what's happening in the heart, but also what's happening in terms of recruiting other immune cells to the heart? So again, collaborating with Paul Fedak and sort of this ongoing sort of partnership is I've been able to work with another talented cardiac surgery resident, Dr. Vishnu Vizanthan, who's also completing his PhD in the lab. And his project really focuses on sort of this epicardial infarct repair sort of concept that the Fedak lab has really been a leader in the field in. And this involves using sort of a bioactive biomaterial that's stitched onto the outside of the heart to provide sort of bioactive components to promote healing. And one of them being primarily a provasculogenic or a proangiogenic sort of effect. And so the Fedak lab has done extensive work looking at different animal models from rodent to pig. And Paul has also done the first in man studies implanting these patches in actual patients undergoing CABG procedures. And what he's shown is that even in this early clinical data is that there seems to be potential benefits in regional perfusion. So indicating that there is a real potential clinical benefit in there. And so I think they're pursuing potentially doing a more extensive sort of clinical trial, but that's somewhere down the line. But what we want to know is, and what he was interested in was understanding some more of the mechanisms of how this patch is actually working. So we know the outcome that there's more vasculogenesis, but how are we actually getting there? So how is the biomaterial actually informing this? So working with Vishnu is that we've adapted this model to the mouse system to actually look at some of the immune components and how it can affect. And so what we found is that with the biomaterial, you are providing improvements in function. So this is just one example with, again, the end diastolic pressure volume relationship. So you're getting a less stiff heart. And similar to what they showed in some of their other models is that we get extensive sort of neoangiogenesis happening. And so this is just a cross section of the heart. You can see the sort of the hollow region here. That's actually the biomaterial that doesn't stain for any of these markers, but it's surrounded, what I hope you can see is surrounded by a halo of green. And this green is actually new vasculature that's actually developing as a result of this implantation. And so we're wondering sort of what drives this sort of angiogenic response. And their group has done work looking at fibroblasts, how they contribute to this, but there's a lot of studies looking at how immune cells can actually also dictate this process. And so what we did is we looked at all of the immune cells that are recruited to the heart sort of post-MI. And what we did is we split them up either in animals that had gotten MI alone or MI with the biomaterial implanted on the epicardial surface. And what we found among many populations that were different under these cases, one that really popped out were a population of innate immune cells called neutrophils that, along with monocytes, are early recruiters to the site of injury. And what was also interesting is we found within this population is there's a subset of them called proangiogenic neutrophils that are known to have a proangiogenic effect. And they're virtually non-existent in sort of the MI alone group, but in the combination with the biomaterial, you actually get these. And what I'm not showing here is that if you actually work up the biomaterial itself, is that you actually see a further enrichment of these proangiogenic neutrophils on the biomaterial itself. And so if you look at where these cells primarily localize, there are some that are found within the infarcted region. So this is the infarcted region here. But if we zoom in, we see that there's an enrichment of these cells along the actual biomaterial border, coinciding exactly with those proangiogenic effects. And if you take a more live look at this and actually look at these cells in action, so here's the biomaterial in white and the green are these neutrophils, is that you can see that many of these cells are actually actively sort of migrating across and directly actively sort of interacting with the biomaterial itself. And so we're sort of trying to understand sort of what are the factors that are being made by these neutrophils. So we know that they're heavy in MMP9, which is known to be very proangiogenic. And so we're going to be testing this more regionally to see if they're enhancing this vasculogenesis through an MMP9 sort of activity. But what this shows you is that by targeting sort of the pericardial space, by implanting this patch on the epicardium, is that we are in fact having an effect on what's happening within the myocardium itself. And so the one drawback of this epicardial infarct repair is that there's some limitations on the patients you can potentially implant it on, unless you go to more minimally invasive procedures. But essentially, it's for patients that are undergoing open heart surgery where you can have a proper access. But then we started to think about, well, what if we could deliver the biomaterial in a different form within the pericardial space alone? So this is some preliminary work that we've been doing where we've actually taken sort of a micronized version of this biomaterial and actually started to inject it into the pericardial space and combining this with our intact pericardial infarct model. And what we've seen thus far, it's still early days, but what we've seen is we're able to mimic some of the functional benefits we see with the epicardial implantation of the patch. So if you just inject it into the pericardial space, we're already starting to see benefits in less LV stiffness And we're pursuing currently how it's affecting the immune response, not only in the myocardium, but also in the pericardial space. And early studies have shown that it can have some pretty dramatic effects. So we're trying to tease that out right now. But it gives us some optimism on maybe being able to help a larger or broader group of patients that are potentially getting a PCI intervention and may be able to be eligible for this type of therapy. Obviously, this is early days, but exciting nonetheless. And so what I've hope I've shown you here over this presentation is the potential impact the pericardial cavity and potentially targeting it can have on the heart, and particularly the injured heart. So whether it be the cells or molecules that are found within the pericardial space under steady state or disease conditions, or alternatively adding exogenous components that can affect this environment. And so what the hope is that by further understanding some of these mechanisms and how this communication actually occurs, we could potentially develop new, in my case, more immunotherapies that target the pericardium and that can hopefully sort of prevent or delay that onset of adverse remodeling that happens in the heart post-MI. So with that, I'll just like to thank the people that have obviously contributed to the work. So the people in my lab, Jameson, Brian, as well as the FEDAC lab have over time become more closely aligned with common interests. So Daryl Belkey, Ali, Vishnu, and Gochi, as well as all their members at large. Obviously the Coombs lab, where a lot of this work initiated in and has served as a good jumping off point for my own lab. And of course, collaborators, Georg Weber at University Klinicum Erlangen, John O'Shea at NIH, and as well as locally here, Karen Poon and the Nicole Perkins Communities Core. And I would be happy to take questions.
Thanks, Justin. That was fascinating, actually. Love the images. I don't know if you were around for Dr. Epstein's rounds a couple of weeks ago.
I was not, unfortunately.
Okay, so he's working on NK cells that kind of are the downstream pathway for all this inflammation.
Yeah.
Do you think inhibiting NK cells would have any role here on the pericardium?
So I mean, I've simplified it to, I guess, basically my interest in macrophages and the innate immune system. NK cells are also part of the innate immune system. So basically, probably every immune cell of of the body will have some impact on this. So NK cells might be involved. So we did see in our human pericardial fluid samples is that we do see two populations of NK cells that are present under different conditions. So what are they doing and whether they're also able to migrate into the heart, we don't really know. But these are something, although we're focused primarily on macrophages in the context of the pericardium at the moment, we're not counting out these other cells. So I think there's way more to explore. Understanding these immune populations within the context of the pericardium is really, I think it's in its infancy, and how they react. Obviously, NK cells, particularly because they can be large sources of a lot of factors that really promote inflammation. And cause a lot of damage. So there actually might be a lot of relevance in pericarditis in particular, but definitely in the myocardium proper, they will have a role. What the connection between the pericardium and that are still to be determined.
So the other thing I heard you mention was MMP9, and I don't know if you're familiar with that or not, but we did some studies in Chagas cardiomyopathy in which we showed that actually MMP2 and 9 we're very closely related with the fibrosis and the promotion and the progression of cardiomyopathy and Chagas, which is a pretty much chronic inflammatory sort of inflammation. So would just targeting MMP2 and 9 be suffice?
So I don't know. I think it depends on the timing of it, and it depends on the disease context. So if in the case of Chagas, you have sort of a continuous sort of activation of that, that obviously could be a way to target it to sort of dampen things down and maybe have less remodeling or adverse remodeling. But what we've seen so far is strategies that have targeted just a simple mechanism usually haven't been enough to have a sustained function. So usually you probably need a multi-pronged approach. The other thing is, beyond just molecules themselves, is actually targeting specific immune cells, either enhancing specific immune cells or preventing their ability to get to the site. Because these immune cells can be the source of many different factors. So whether it just be MMP9, but also other things that promote or drive sort of pathology. And so maybe a single molecule inhibition may be it may be too simplistic. We may need a multi-pronged sort of approach, and maybe even targeting the immune cells themselves that are sources of many of these factors. Maybe also another way to do this. But I think it's very there's some subtlety in there, so I think it's very time dependent. So in the context of post-MI remodeling, for example, you want a certain level of fibrosis to be able to build up that scar and to strengthen up the wall to some extent, but you don't want that fibrosis to necessarily persist for a long period of time where you're getting further effects on damage. So I think an all or nothing strategy is maybe a little bit too simplistic, and I think it's really disease context specific on when you want to provide these and how long you want to provide them for.
Yeah, well, I have another million questions myself, but one of the things that maybe we should talk about is that, in Chagas cardiomyopathy, there's a significant proportion that end up having VT, and the way we ablate that VT is accessing the pericardium. So, if you want to inject some of your thing in Chagas pericardium, just let me know. So
Okay.
There's a few questions here. Do you think monoclonal antibodies have impact on myocardial remodeling similar to GPCMs?
So monoclonal antibodies. So so that's a pretty broad question. So I mean, there has been evidence that monoclonals say in the CANTOS trial that is targeted sort of IL-1 beta targeting has shown some benefit in terms of incidence of cardiac events. In terms of its effect on remodeling, I think there's some ongoing studies right now looking at monoclonals in the context of sort of, especially in the chronic sort of remodeling phase, there's a lot of talk around sort of this low-grade inflammation. And so there's a number of, I think, ongoing clinical studies looking at these monoclonals to see if they can dampen down. So whether it be targeting IL-1 beta, IL-6, TNF alpha, and there's, I think, already approved monoclonals for every one of these applications. So I think there's definitely a place for these. The one caveat I would give is that, and they've shown in the CANTOS, although it's sort of tucked away, is that the patients that receive the monoclonal, they actually have a higher incidence of infection as well. So you target something to dampen down, maybe to target the cardiovascular disease, but a flip side of that is you're affecting the immune system as a whole and its ability to maybe respond to other insults. So the ability to maybe provide these monoclonals maybe more locally, maybe say in the pericardial space, is a sort of an interesting idea to get around that, where you have very low turnover of the pericardial fluid, and it doesn't seem to get out into the circulation and can things that have been injected into the pericardial space can get into the myocardium and be more enriched than delivering it systemically. So that could be sort of an interesting avenue to sort of explore. But I think there's definitely a room for monoclonal antibodies in sort of regulating this. But we have to do it in a very find ways to do it in a more targeted approach.
Okay, so Dr. Howlett thinks that you gave an excellent and provoking talk, and he's wondering if some of these mechanisms may be downstream to neurohormonal activation following injury in humans, or any evidence in animal models for this approach to be tested, in addition to typical medical therapies like beta blockers, aldosterone, et cetera, and whether you're planning to do that. And Todd Anderson has a question following that.
Okay. So sort of the, if I understand the question, sort of the sort of neuroregulation of the immune response, I guess. So this has been sort of a hot topic and has really become at the forefront of the immunology world, is the ability of, or the interaction between sort of neural input in actually regulating how the immune response works. So I haven't looked at that yet. It's something I'm interested in, and I think there will definitely be interplay between those two, particularly in the context of post-injury and sort of how this proceeds. Now, whether these neurohormonal sort of regulation is different in the pericardial space and in the myocardium itself, there's probably differences there, but yeah, it's on my radar, but I haven't, we're taking small steps at the moment. So
I guess, way back when I was a physiologist, we did some models in which we looked, because you're doing some transmural myocardial infarction. So there's differential innervation between sympathetic, endo, and epicardial, as well as vagal.
Yeah.
And, vagal modulation. So what we were trying to do way back then was trying to modulate the vagal to A, increase angiogenesis, and B, decrease inflammation. So those can be. Yeah. So Todd.
Hey, Justin, nice talk. Nice to see you. So some recent clinical trials in both chronic and acute coronary syndromes / MI have shown benefit for colchicine. And so this has led to the approval of colchicine as an adjunct therapy infarction with pretty striking reduction in events. So I guess my question for you is there anything to suggest in the mechanism of action of colchicine as an anti-inflammatory that the pathways you discussed today could be at play with that particular therapy?
Yeah, I mean, there's definitely, I mean, anytime you're using sort of a broad approach, there's definitely going to have an impact on your immune response or your immune cells more globally. We haven't looked at that just yet. And again, we're looking at a very sort of controlled system where we're with lack of comorbidities and also lack of therapeutics that are on board. But as we're understanding more about these cells and how they contribute to the problem or to the fix of the problem, we want to better understand how these different therapeutics that are approved clinically, how they interact with these cells, obviously. So it could be that they're dampening down very acute inflammation while maintaining sort of some of these homeostatic mechanisms like pericardial macrophage. So obviously something we want to look at. We have to start somewhere, and yeah, definitely worth pursuing though.
Well, that's a good segue to announce that Dr. Tardif, who was one of the PIs of the Colcott trial, is going to be presenting rounds November 9th, and he's going to be reviewing the topic of inflammation, colchicine, and all that. So make sure you tune up. Justin, thanks. That was fascinating, and appreciate you having done this for us. And maybe we should talk about the Chagas thing and some human stuff, and actually some animal models that we were working, also looking at some of the pericardial inflammation and myocardial inflammation in a mouse model we have down there.
Yeah, I would be happy to.
Okay, well, thank you very much.